decades now, everyone wanted to find out how we can stop, slow, or prevent Parkinson's disease. And that has not been possible by a long shot. There have been animal models that came and went, breakthrough promised very high potential candidate drugs that came and went. And I'd like to discuss as to what went wrong there, what we think today, why none of these actually worked and translated to human use, or why they failed and weren't successful in human clinical trials. Where we are now, what we are doing now in this, and why do I think that the future is much more truly promising finally. And there is some true actual breakthroughs and advances that are happening these days that make all of us much more hopeful and excited. So my personal interest in movement disorders started way back, and soon after I was working with one of those early animal models, and I was very interested in neuroprotection and disease modification early on in my career. During my postdoc years, I took 2 years off after medical school when I worked on these animal models. And then throughout my career, I was working with disease-modifying clinical trials, as well as various biomarkers for measuring disease modification treatment effect, as well as progression in general in Parkinson's disease. And I'm happy to share with the group some resources later as there is interest in it. So when Parkinson's disease was originally understood from the pathological standpoint, it was clear that certain neurons were dying at a much greater rate and with some selectivity than other neurons. And the focus went down to the nigrostriatal or dopaminergic neurons. And in the late 70s and early 80s, it was discovered that a certain toxin, which was contaminating homemade street drugs, actually made people overnight severely Parkinsonian. And that was the MPTP story, which really changed the way we thought about Parkinson's disease modeling. And there was a now discovered, or now we understand it was is a false hope or fallacy, whereby it was assumed that this toxin, the MPTP, that caused severe Parkinsonian state in humans, can give us animal models where we can understand how Parkinson's disease starts, what causes it, and also how we can prevent it, how we can intervene the process. So animal models were set up, were founded with the use of MPTP in many different species, starting from mice, even up to monkeys, primates, where MPTP was administered in one way or another. And these animals became severely Parkinsonian. And then a number of different agents, drug candidates, were tried in those animal models to see if the Parkinsonism can be prevented, mitigated, slowed. What happened was several different such candidates and agents and drugs were in fact identified in those animal models, which were profoundly effective in preventing the MPTP-caused form of Parkinsonism in these animals. And there was a lot of excitement that now we found the Holy Grail, we found something that can prevent Parkinson's. Now, it turns out that all those different agents that were tried in those animal models and looked very promising and very successful, when tried in human clinical trials, unfortunately, all of them failed. And none of them, not a single one of them, translated into any degree of success in human clinical trials. In other words, they were either not usable in humans because of diminished bioavailability, not penetrating the blood-brain barrier, or toxicity on their own, so that ruled out any form of meaningful clinical use, or when they were applicable and usable in humans, they were ineffective. They were ineffectual. And unfortunately, that lack of translation and that sad and depressing story of lack of any success with disease-modifying and neuroprotective treatments continued for decades, more than 2 decades to be exact. And people were very despondent, very depressed as to whether we will ever crack it, whether we will ever be able to find some cures or some treatments that can slow, prevent, or mitigate the Parkinson pathology. All the while, we were very successful in bringing symptomatic treatments to the clinical treatments armamentarium. And levodopa was invented in the late 60s and then started their success in the early 70s. Levodopa, when discovered, was a tremendous game changer, turning patients who could barely move into nearly normal function. And I think if you can look up the movie Awakenings with Robert De Niro and Robin Williams, ironically, Robin Williams just died of Lewy body dementia, which is a related disorder. But in that movie, he played Oliver Sacks, who is a famous neurologist. And one of the greatest story was his discovery of levodopa in this rare Parkinsonian condition called phonoeconomal disease or encephalitis lethargica, where this levodopa made a miracle-like transformation in those patients. So that story continued, and many other symptomatic treatments were subsequently added, including dopamine agonists and many others. And then with the advent of deep brain stimulation, we really have a very strong list of treatment options that can symptomatically help patients. And all the while, we remained without any disease-modifying or neuroprotective treatments out there. So the MPTP story, unfortunately, caused even Congress to question whether any more hundreds of millions of dollars that were spent on developing these animal models and doing the clinical testing with these what looked like promising Parkinson's disease progression slowing drug candidates, whether we should put some more good money after bad money, or whether we should just look for a different direction. More recently, it was recognized that part of the reason why all of this translation from the MPTP and other toxin-based, because it's not just MPTP, that's the most famous one, that's the most well-known one, but there are many other toxins that have been identified that can make animals Parkinsonian, and it can also make humans Parkinsonian, just like MPTP did. But this Parkinsonism that these toxins produce is not actually the same as the idiopathic Parkinson's disease that develops in most people. And therefore, any results that we gathered from these animal models was not something that we could extend, expand, and extrapolate to human actual Parkinson's disease. And that was one of the problems. The other problem for why we think these translations failed so spectacularly was because we have the complete control in animal models of timing. You can prevent and protect those animals at any given time before or after the insult. So in other words, you put MPTP into the animal, and then they will suffer the injury of their brain causing Parkinsonism. And then when you apply a preventative or protective agent, you can do that before they are exposed to MPTP, and then it will prevent it. But you don't have that timing control in actual patients. When they develop Parkinson's disease, they already have the symptoms when they see a doctor. So you can't roll back time and then apply some kind of a protective measure or a procedure that could prevent their Parkinson's in the first place. And the third reason why we think these previous attempts failed was because it did not account for the fact that Parkinson's disease is very diverse. Not every patient has the same cause, the same mechanics, and the same molecular pathology that leads to Parkinson's. And those individual differences may be very, very critical in how these disease-modifying treatments work in some, but not all patients. In other words, our entire clinical trial experience in Parkinson's disease was largely based on the symptomatic treatments, such as levodopa and other dopaminergic medications primarily. And those experiences and that type of setup was not applicable to disease-modifying treatments. Patients with Parkinson's disease, for the most part, will respond to symptomatic treatments similarly, or at least largely similarly, not entirely, but they all have dopaminergic deficits. And so if you apply a dopaminergic agent, they will get better. So our clinical trial networks were based on that assumption that patients behave similarly in these clinical trials. And so we can have a lot of them, and then we can control for various compounds by large numbers. Statistically speaking, we will get the answer whether a drug is effective or not by doing it so. Now, in disease-modifying treatments, it may not be exactly like that, because some patients could be responding to a certain treatment. Other Parkinson patients could be very different from this perspective. So with all that learned now, I think the reason why I mentioned breakthrough is because more recently, we are looking at a number of different animal models that are far better than the MPTP or other toxin-based animal models. We no longer just inject some kind of toxin or poison that will knock out all of these dopaminergic neurons in the animal overnight, because we know that that's not how Parkinson's disease starts. That's not very informative about the inception of disease. If we want to interfere with the underlying molecular process, we should be able to model that accurately and much better than just knocking out all those cells, because the way that those cells die in a toxin-based model is not how they die in an actual patient. This is fundamentally different, and that difference is going to account for the failure of any result from these toxin-based animal models to actual patients. Instead, we are looking at animal models that represent the human disease much better. So what helped us establish those new generation of animal models is largely our better understanding of what's happening in actual patients. We understand the pathology, the pathophysiology much better now. We found a particular protein called alpha-synuclein, and it has been identified as very central to how the disease starts. What actually happens that kills those neurons in the brain in actual patients with Parkinson's disease. Turns out this protein called alpha-synuclein is something that we all have. There's a normal version of it that we don't quite understand the function entirely in a normal person, but certainly it is good for something, or it does something that we need, because it is present in everyone. Now, what happens in Parkinson's disease, this protein starts misbehaving. This protein acts in an abnormal way, and it misfolds. This term of misfolding refers to the structure of this protein. This protein, just like any other molecule within the cells, usually is soluble. It's within the cellular fluid, and it is in a solute form. Now, when this particular protein, the alpha-synuclein, changes its conformation, it becomes no longer soluble. And so when it is no longer soluble, it will aggregate. And as it aggregates, it becomes toxic. And it will form the well-known Lewy bodies, which are little inclusions in the cell made up by this bad synuclein. Now, since we understood that, we have identified a rare but important genetic form of human Parkinson's, where we see a mutation in the gene that transcribes this particular protein, the alpha-synuclein. When you have this mutation, then your alpha-synuclein will misfold, and then it will propagate through the entire brain and will cause Parkinson's disease. What happened was we are now able to take that human gene, the bad human gene for alpha-synuclein, out of patient's genome and put it into a mouse genome and make the mice carry that abnormal human gene. And these mice will transcribe that alpha-synuclein, the bad one, and will become Parkinsonian. The process that they will become Parkinsonian will mimic the human disease very accurately. And why that is very important is because now we have animal models that resemble the actual human disease far better from the perspective of how the disease starts, how these neurons begin to die. There is yet another animal model, which is not transgenic, but which is based on injecting these fibrils of this alpha-synuclein, the bad one. So you can isolate those little fragments of the bad alpha-synuclein molecule, and those are referred to as fibrils. And protein fibrils may be protein fibrils fragments, so it's abbreviated as PFF. These animals are perfectly normal mice. They are perfectly healthy, but when you inject these alpha-synuclein bad fibrils into their brain, it will turn them into Parkinsonian based on bad alpha-synuclein, just like in human disease. Now, what we gained with these animal models is a much, much better platform for us to test any sort of agents that may, in fact, slow down, prevent Parkinson's disease. So this is a relatively recent development. These new generation of animal models have become usable and available only recently. But even this relatively short period of time, we have identified a large number of molecules that seem to be very effective in these animal models. I mentioned the timing before, and I said that all those preventative treatments from the MPTP animal models were largely useless because you could just apply the preventative intervention before you apply the toxin, and voila, you prevent it, but it's not practically applicable in actual patients because we don't have a time machine. Now, what happens with these new animal models and the new agents that are being tested in them is that these animals will already have the underpinning, the underlying causes of Parkinson's disease. The process will be already ongoing when you test a disease-modifying or neuroprotective intervention. And then if it is effective, it's not like you have this completely impossible and practically irrelevant timing issue. You are timing your intervention in a way that would be applicable to a human disease because the underlying pathology is already ongoing, and that's when you apply this potential neuroprotective intervention, just like in real disease, just like in the actual Parkinson's disease in humans. Having said that, for the first time last year, one particular agent was in fact found to be effective in human patients. The agent is called exenatide. It is a diabetes drug that's already on the market. One of the trade names of it is glyburide. This exenatide is one currently available medication that is, I'm sorry, I misspoke about glyburide. The brand name I wanted to say is Bydureon. Bydureon is the trade name of it. So it's available for diabetes now. It's not approved for Parkinson's, so we cannot prescribe it for Parkinson's. But this is the first drug that was found, at least in one study, and it was just a one-site study. The first neuroprotective or disease-modifying clinical trial published in Lancet last year from the UK, where there was a statistically significant benefit in slowing down the progression of Parkinson's disease. So it's a small result, it's one trial, it's just a one-site study. There are many problems with this study, but still, it is a breakthrough from the sense that we have never been able to point to any clinical trials ever that actually was effective in terms of disease slowing. I would also say among the reasons why we failed before was in a great part due to the fact that we didn't have very good outcome measures. We don't have a good biomarker to tell how severe the Parkinson's disease is and how we can measure whether one of these disease-slowing, neuroprotective, or disease-modifying treatments are in fact effective. We can't count the brain cells at the moment, and there are many different ways that we are looking at improving our outcome measures to improve our ability to detect disease modification or detect the ability to slow down the disease progression. Right now, we are still reliant on the standard methods with hand grips, with bradykinesia clinically assess the rigidity, the severity of tremor or walking impairment. All of these things are still the gold standard to look at severity of Parkinson's disease. But of course, these measures are subjective. They are affected by symptomatic treatment. So they are very limited for our use specifically for telling if a disease-modifying therapy is helping or not. So there have been some major advances also in this area, in the measuring of the Parkinson's disease severity, the measuring of disease progression, as well as the effect of disease-modifying treatments. And these are increasingly being applied in our current new clinical trial designs. And there are many current clinical trials ongoing that are specifically for disease-modifying agents, and several of those already are testing compounds or candidate drugs that have been found to work in these much improved new generation animal models that I described. So that's why we are so hopeful. Again, we have much better animal models now. We have the first clinical trial that actually showed a little bit of effect in terms of slowing down Parkinson's progression. And now we have better ways of measuring Parkinson's disease. So all that together makes me predict that we will have, within the next 5 years, a effective disease-modifying agent in Parkinson's disease that will be proven to be effective in a large clinical trial, such as a phase 3 multi-center double-blind clinical trial. I think I finished exactly on 30, so I'm happy to take questions.
Great, thank you. You know, I have not heard the history and the past about, you know, Parkinson's research. So thank you, that was so interesting, and it's so important to see the full picture. Can you see Bart's question in the chat? Yes,
yes. In fact, I was an investigator on the PPMI while I was at Johns Hopkins. We were one of the earliest sites in that, and that effort is specifically to find out progression markers initiative, progression markers for Parkinson's disease. So if you see how much money has been spent on that effort, one of the candidates in that is actually the DET scan, which is currently approved already for diagnostic confirmation of Parkinson's disease. But in the PPMI, we are working on trying to find out whether the DET scan can also provide some quantitative rather than just qualitative data with regards to the severity of the disease. So what I'm doing now is I'm going to send you a link here where I discuss that in greater detail as a PPMI investigator while I still was at Hopkins.
Thank you.
So the question is, how do we, oh, okay, there is another question. Can you spell the diabetic drug? Yes, this is exenatide. So the next question is whether the short duration of the mouse life is a factor in terms of how good these animal models are. In fact, that's an advantage, because that way we can evaluate things in an accelerated manner. So mice live shorter, but everything is happening quicker, and so in terms of the duration of these changes, it is a good model. Again, the earlier models where you instantly wipe out a certain neuronal population is not a good model, because that's not what happens in real life. What happens in real life is a slow process where neurons are declining. Not only that they are declining, but they are declining in a particular pattern and a particular order, and slow, what we call is propagation. So the synucleinopathy, or the pathology that involves synuclein, starts in one point in the brain, actually 2 points, in the olfactory bulb and down in the lower brainstem. And then it kind of spreads slowly through the brain. That spreading, that pattern that mimics or emulates the human disease is in fact happening in these newer animal models. It was not happening in the toxin-based models at all. In the toxin-based models, basically, even if it happens slowly, because they tried to make the toxin-based models such that they don't use a high dose of the toxin to knock out all of the dopaminergic neurons, instead giving a little bit and then a little bit next day, and some of them spread out over 6 weeks. But still, it has nothing to do with the actual human process, in which it starts in one point in the brain and slowly spreads. Not only that it slowly spreads, but not every neuron is equally susceptible to this. For instance, the nigrostriatal dopaminergic neurons are very susceptible. They go early and they go at a high number, and they are lost rapidly. But that doesn't mean that other neurons are completely preserved. It's just a particular ratio and a particular rate that these neurons get taken down. Now that has to do what the clinical manifestations of the disease is. So you have to well emulate that if you want to truly model the human disease. And in these new animal models, that is happening. So that's another reason why we think they are very, very good models. And in terms of it happens in a shorter period of time, well, the mice live shorter. So in their life, 1 month is representative of 5 years in a human. So we get to evaluate these changes as well as the disease modification in a scale that we can work with, because we can't wait 20 years to answer a question about this. So we have this as an advantage, and that's why we use mice as opposed to primates. But of course, once we get some really good treatment or candidate or promising results, then we will take it to primates, and we will do that. Can you spell the diabetic drug? Yes, so I did that. Any thoughts about the effectiveness of exercise and eating and a low inflammatory diet? So first of all, let me comment on exercise. So besides all the drugs that we are testing and looking at to slow down disease progression, without a doubt, exercise is a very important way of doing just that. Now, how effective it is and what the mechanism of the effect is not entirely known. Also not entirely known what exercise kinds or types versus other types may be more effective. So what we do know is that people who exercise, and I emphasize on a regular basis, will generally do better over time. So if you look at a patient with Parkinson's disease who exercises on a daily basis for the next 10 years, 10 years later, they will be more mobile, they will be more functional, and they will have a lower motor score, a lower Parkinsonism motor severity than their counterpart who maybe having the same severity of Parkinson's disease at start, but did not exercise for over 10 years. So 10 years out, the person with exercise will look better, will be better. Now, we don't know exactly whether that is because we are protecting neurons, because the exercise releases some chemicals that are anti-inflammatory, because the exercise suppresses some cytokines that are pro-inflammatory. So we don't know if there is an actual molecular underlying mechanism for the disease-modifying effect of exercise, or it is purely mechanical. Some people just attribute that to so-called conditioning. So if you don't do much, if you don't exercise, if you're not doing as much as you can, then gradually you get used to that. You get used to doing less and less, and that accumulates over time, and you accept that you can't get up as much or walk as much, and if you don't exercise, you will be in a worse shape just because of that so-called deconditioning effect. Now, whether the exercise's benefit is mainly due to the avoidance of the deconditioning effect and is more like a conditioning, your muscles are in shape, your motor circuits are practiced, and you are habitually ready to do more, or is there an actual neuroprotective effect? Nobody knows at this moment. Now, the second part of the question was about diet. So we know even less about diet. There have been several attempts in trying to identify anti-inflammatory vitamins and antioxidants and all sorts of things. Nothing firm or confirmed or reproducible so far has emerged out of that. So there's some theoretical reason or basis to think that if you have an anti-inflammatory or antioxidant in your diet, then that will help slow down the progression, because neuroinflammation is in fact an important part in the cellular death. But at the same time, the naysayers will point out that in a normal, regular North American diet, with fruits and vegetables at a normal rate and sort of what your normal diet is, it's almost impossible that you wouldn't get a saturating level of those vitamins already or those antioxidants already. So adding more on top of your normal regular diet because of the ceiling effect may not have a great potential to change anything. And also it could be that the brain buffers these. So again, there's not a whole lot of evidence for efficacy there. Obviously, we all benefit from eating healthy and also exercising. So I wouldn't sort of try to eat a lot of supplements and vitamins and or just have some reasonable expectations in terms of whether they do anything good. So the next question is about isradipine, and it's not just the Northwestern. Northwestern was the headquarters for the study, but in fact, it was a multi-site a placebo-controlled trial, which was a large trial, recently closed enrollment. I was an investigator for that, both at Hopkins and then after I moved here to Cleveland Clinic in Las Vegas. And so the trial name, if you want to look it up, is called Steady PD 3, because it was a phase 3. And that was something that recently was closed to enrollment. So we don't have the results of that. We are still awaiting the final analysis. So this study was based on the observation that our nigrostriatal dopaminergic neurons burn energy at a high rate. And that high burn energy is what contributes to their susceptibility and being vulnerable to any form of damage, including one that comes from alpha-synuclein. And why are they burning energy so much? That's because they have a constant depolarization of their membrane. There are so-called leaky calcium channels in them. You have to know that calcium in the brain is such that its concentration within the cell is thousands of times below what it is in the extracellular fluid space. So there's a tremendous gradient for the calcium trying to enter the cell. When it does enter, you need a lot of energy to pump it out against its concentration gradient, and that takes a lot of energy. So this study was based on the assumption that if we block those calcium channels, then there will be less of an influx of the calcium into the cell, and that will be unburdening the nigrodopaminergic cells from this high energy use, high energy demand state, which contributes to their vulnerability or susceptibility. So that's a concept that's apart from the synuclein-based approaches. It's a good idea. Now, whether it's going to actually show benefit or not, we will have to find out once the study results undergo the final analysis.
Okay, and Dr. Mari, someone shared a question with me. I'll read it. I don't understand a lot of the science that you're discussing, but it sounds impressive. What do you tell your patients about hope on the horizon?
So I am very excited about the hope in the horizon. What I ended with all the discussion about, one, much improved, truly representative animal models, two, better ways of measuring Parkinson's disease. It's not the DET scan that somebody asked about the PPMI necessarily, although that is still ongoing, but there are measures like we can have wearables that can track your bradykinesia over a week or longer. We have many other devices that can objectively assess you, not just one time point. So again, we are better in animal models. We are better in measuring Parkinson's. And so we are also better in doing clinical trials specifically for disease modifying. So all of these actually led to the Steady PD that we discussed is one trial that has just closed and undergoing final analysis. But there are two other major phase 3 trials that are either closing or undergoing now. And there are many molecules in the pipeline that are even more promising. Not only that, but there are at least 3 or 4 targets. Targets meaning how you're going to interfere with that abnormal process, the pathology that brings these cells down. And then we have all of a sudden an abundance of these targets that are brand new. 3 of these targets were not known 5 years ago. And these are the most promising targets where we actually have a handle in preventing cellular death, helping the patients with Parkinson's disease enjoy a longer life without progression. I would add to that when I mentioned that symptomatic treatments are very effective, it's true with a little bit of a caveat. They are effective for motor symptoms, and not all motor symptoms, but some motor symptoms, which means that in the early disease state, when somebody has a little bradykinesia, which is slowing of movement, maybe tremor, maybe stiffness, we give them levodopa or any one of these dopaminergic drugs, and they will be markedly better. It will help a lot. And if it's done right and the medications are optimized, we can make a big difference in their lives. But later in disease, these problems will worsen. These medications do not stop the progression. And then usually balance problems, postural problems will develop. And those problems are not as responsive to these dopaminergic agents anymore. And then non-motor problems also develop, not because of the dopaminergic neurons that are still dying, but you only have so many of them, but this is other neurons, other systems that are responsible for cognition, for mood, for sleep, and so on. And we don't have too many useful treatments for problems affecting these other networks that get affected by Parkinson's disease as well, besides just the dopaminergic networks. Now, with the disease-modifying idea, we protect all of those neurons. It is not just the motor symptoms, not just the dopamine-related problems, but we are also hoping, and this has a realistic expectation for protecting every part of the brain, including these networks that are responsible for the non-motor issues, such as cognitive impairment and others. So I tell my patients that finally there is hope. I think it's very realistic to expect, as I said earlier, that within 5 years, we will have a breakthrough. We will have a large phase 3 placebo-controlled trial that will, for the first time, not fail like every other in the past 20 years.
Wow, that's
I could not
great.
say that 5 years ago. I'm not like a, reckless, lunatic who will say anything just to be positive. That's not true. These are statements that are largely agreed upon among experts and based on data.
Okay. And I remember I hear a lot how the Michael J. Fox Foundation is actually spending more money on Parkinson's research than the federal government. I mean, it seems like we're at a very exciting point. Why do you think that the government isn't investing in this research?
Well, you have to bring somebody else
Okay. Unfair question.
From Congress. So basically, there are several elements to that question. And one, I think, as I kind of alluded to earlier, is that this is an area where science, unfortunately, has not really produced a lot of results. And every time they said they would. So Congress got a little, wary of that. And it's like, we're not throwing good money after bad, except now we really have a good story. So, but I think the other part of this is that NIH funding has stagnated in general, not just neuroscience, not just Parkinson's. In general, neuroscience probably had it a little worse, a bit out of proportion relative to the entire government funding for science or NIH budget, but everybody suffered, and it started up to the Iraq War. There was a very steady increase of NIH budgets year over, and because we went into the war, that needed money, and then that state just sort of fixated ever since. And overall, I think it's a societal question you're raising. How much should we as a society, as a country, through our representatives, through our Congress, prioritize spending in science versus on military, versus on other causes, versus on tax cuts? So obviously, if you have massive tax cuts to corporations, and this is a political issue, then that money is not going to go to public spending, such as research funding. And actually, that's the narrative of Republicans that why isn't the donation and charity sector take care of that? They don't think it's the government's job.
Okay, I guess you're right. That wasn't a fair question to you, but it's.
I would add that it's not just the Michael J. Fox Foundation. They are doing an outstanding job. However, the Parkinson Foundation needs to be mentioned because as much as the Michael J. Fox Foundation has long focused on these exact studies that I described, the so-called disease-modifying ideas, as well as the proper biomarkers for them. The importance of Parkinson Foundation funding needs to be added. They are not necessarily focusing solely on that, but they have this network of the centers of excellence. And that is what can help patients everywhere, all of them. Only a few patients will qualify for these disease-modifying trials because the inclusion-exclusion criteria will be very, very restrictive. We are enrolling now for one of them called NILO-PD. I'm going to write it here, which is one of the high-profile research trials that currently is enrolling. But we have failed. We pre-screened 48 patients who were potentially candidates. 6 of them passed the pre-screen, and only one of them, who I'm going to see right after this, by the way, for the screening visit, reached the screening. So it's very hard to enroll in the studies. On the other hand, the Parkinson Foundation brings great care to everyone who is able to go to one of the centers of excellence. And they also do a lot of research in finding out how we can optimize care in Parkinson's using our current tools. So it's one thing that we are all excited about developing disease-modifying, futuristic, almost sci-fi treatments which have the potential of not just slowing but maybe preventing Parkinson's disease altogether. But the other important part we have to recognize is that we have patients today who need great care, and so that is also a very important part that's largely funded by the Parkinson Foundation.